My name is Andrew Basaga, and I have been a junior board member of MIB since 2018. I was diagnosed in 2017 with osteosarcoma in my proximal femur, undergoing my chemotherapy treatment at Advocate Children's Hospital in Oak Lawn, Illinois, and receiving my limb salvaging surgery at Rush University Hospital in Chicago by Dr. Gatellis and Dr. Alan Blank. I will be 4 years NED this July. I am here to cause a cure because, as a survivor of osteosarcoma, I feel that every child should have as fortunate an outcome as I. I'm pleased to introduce Dr. Gorlick, who will be moderating the Big Data and Biobanks panel. Dr. Richard Gorlick is Division Head and Department Chair of Pediatrics, Department Chair ad interim of Sarcoma Medical Oncology in the Division of Cancer Medicine at the MD Anderson Cancer Center, and H. Grant Taylor, MD, W.W. Suto, MD, and Margaret P. Sullivan, MD, Distinguished Chair in Pediatrics. His laboratory is the founding bone tumor resource laboratory for the Children's Oncology Group. His molecular pharmacology laboratory is completely focused on osteosarcoma. His laboratory is a member of the NCI-funded Pediatric Preclinical In Vivo Testing Program and past member of the Pediatric Preclinical Testing Consortium. Dr. Gorlick is involved in clinical trials in part as past chair of the Bone Tumor Disease Committee for the Childhood Oncology Group. He is a past president of the Connective Tissue Oncology Society and is current chair of the Cancer Prevention and Research Institute of Texas Advisory Committee on Childhood Cancers. Dr. Gorlick has published more than 304 peer-reviewed papers, reviews, and book chapters. Please welcome Dr. Gorlick.
So thank you all for being here. Appreciate the introduction, appreciate the organizers putting together this meeting, and Christina and Anne inviting me to sort of serve as the, panel lead for this group. As usual, whenever I do something, I'm trying to advance the slides. Can we start getting a little bit forward? Okay, beautiful. We're going to skip that one. So, I see him here. Hopefully those slides are going to show, but I'm going to keep speaking anyway. So basically, I'm going to present this panel a little bit differently from everybody else. So every other group goes one by one and goes in detail through the sort of panel members. This is an immensely distinguished and large panel. And if I had to look at any one of their single CV, I can spend the entire 5 minutes reading it. So instead, what I try to do is figure out how am I going to sum them up very quickly. So I looked for common elements, and I started with geography. And geography doesn't work at all. Like Sam's in Chicago, Brian and Corey are in Boston, Elena's in Santa Cruz, Patrick's in Texas, and Beth is in St. Jude. So that didn't work at all. I looked at education, and education was just as bad. Even though Boston Children's is sort of the modal program for these groups, there's a lot of heterogeneity there. And we have folks here, and it's not on this part of the slide, but we have MDs, we have PhDs, we have MD PhDs, so that didn't work. And I said, this is a great introduction, because the panel's heterogeneous, and so is osteosarcoma. That works. And osteosarcoma is all about bone. So what's common about this group? What's common about this group is every single one of them leads an incredible initiative that's large and we're going to hear about today. So whether it's hearing about Data Commons, whether it's hearing about Project Every Child or the Osteosarcoma Explorer, whether we're going to hear about Count Me In, because I'm not going to call that the Osteosarcoma Project, or we're going to hear about the Treehouse Initiative, or we're going to hear about sort of everything Patrick has done as sort of the Cancer Center Director, or Beth through the Childhood Solid Tumor Network, let alone not mentioning Pivot or other things she's part of it. We're just going to sort of start with that. And then I figured, like I have, all right, the big green button, not the little green button. I figured, instead I'm going to have like 2.5 minutes. So that's going to allow me to give my impression of what we're talking about in this panel. So I made this slide all by myself. You can see it from the complexity of the slide. I didn't have anybody help me with this, but I did steal most of the clip art from the internet and probably violated copyrights, but who knows. And really what we're talking about is what we get from patients, and to be honest, there's 3 little people at the bottom. That's the rest of their family. And what we get from patients is we have tremendous amounts of information and medical records. We're able to get samples from them, like blood or buccal swabs or swabs from other places. And that little thing, red thing, is sort of meant to epitomize their tumors. We can process them through machines, and we get to things like data or biomaterials. Why is this so important? The reason this is so important is because these resources aren't consumable. And as long as they're shared to everyone possible, the amount of utilization is possibly infinite. Critical is getting as much information as possible. And there's a lot of requirements to make this work, and I'm sure we're going to hear about this as the talks go on. So I want you to think about a couple of things as we think about these talks. Some of them are, a lot of these are going to offer direct participation or the ability for your doctors to plug into them. I hope you think about whether you want to talk to your doctors about whether you want to enroll in these things, whether you think you want to enroll on them, whether you want to use them. And like all things, there's risk and benefit. Obviously, you have to make the decision to participate or not to participate. There's benefits to the community and maybe yourself. But at the same time, there's always things that are a little bit more difficult. And so I'm going to end right on time, heading to each of the panelists. I'm going to have, they're obviously going to continue the introduction, so if anything was confusing, they'll tell you actually where they're from and who they are. And I look forward to each one of their talks. It's the big green button. And welcome, Sam.
All right, thank you. All right, hold on tight because you're about to get a 20-minute talk in 10 minutes. If you look at the bottom of every single slide, there's a link, and that link will take you to my slides, an expanded version of the slides that includes all the ones I had to cut out to try to squeeze this in. And I would encourage you to take a look because there's some more explanatory slides in there. We're talking about data commons today. Data commons are not databases, they're not registries. Data commons are cloud-based areas where you can put data and you can put tools and you can put information and you can analyze it all in one place. Ultimately, data commons are here to help you not have to download the data to your own computer, not have to build your own pipelines. The dream of the data commons is that, and remember, a cloud is just somebody else's computer. The dream of the data commons is that there will be a place to put all of our data and tools and to obviate the need for having to build these entire server farms at every institution. So today, think about data commons. Now, the Pediatric Cancer Data Commons, which I've been working on for the last 6 or 7 years, is a hub for all pediatric cancer specialties. So we want to provide cross-disease research. We want researchers to come in and say, show me all the solid tumors with an ALK mutation. Show me all the kids who were treated with doxorubicin who survived greater than 5 years. We don't want to limit ourselves to the traditional silos. And our secret sauce that we work on is the development of a common data dictionary that I'll talk about. I'll talk about this exciting area of data standards. We work on an international level, so we have people from all over the world contributing to our data commons, and we really take responsibility for education and data literacy. We want people to learn how to develop better ways to collect data, how to develop better ways to share data. Now, we deal in clinical data, mostly demographics, lab values, tumor information. We will take genetic data or genomic data or imaging data if we have to, but we would much rather link to those data. Those data are much better served by those who specialize in those data. There's a genomic data commons, and we should link to it. We have been working on completed clinical trials, but more and more we're starting to get data right out of the electronic health record. And of course, we've been working on pediatrics and AYA, and we're even moving into now using adult data. So this is, in a nutshell, what we're dealing with when it comes to standards. People can choose whatever label they want when they collect data. And so one hospital might use a 0 or a 1 or an M or an F when they're talking about biologic sex. And that creates a lot of confusion when you're trying to combine data sets. So our group comes in and our group gets consensus. And we don't just get consensus with 1 or 2 groups. We try to get worldwide consensus. We bring a big group together and we try to develop a consensus way to describe any particular area. And in this case, we could decide on these 3 words for biologic sex, but then we link to a code set in the NCI called the NCI thesaurus. Now there's no more ambiguity. That code stands for male. You don't have to worry if it's misspelled, if it's lowercase, if somebody put an M. You have the code and now it's unambiguous. So now what we really go after are these consensus data dictionaries. And if we take a closer look at one of our completed dictionaries, you can see that here's the consensus we got from the osteosarcoma community on medical history. And it was not easy to come to this consensus. The people in the US and Europe, they all wanted different things on this list. We came to a consensus that this would be the list of conditions we would include. And again, everything is linked to something in the NCI thesaurus so that you have to worry. I can't spell half the words on that slide, so now that we have a code, we don't have to worry about somebody misspelling it in a data collection form and then having trouble combining it. And if we need to expand this list, easy. We have a whole change management process for expanding and making the list different. We have about 500 lines in our leukemia dictionary and our solid tumor dictionaries. They're very big. They're all open source, available on our website, and I would encourage you to look at the dictionaries. You can share them. They can be used for other purposes. Everything we do, from the code we write to the dictionaries we build, is all given back to the community. We are also working on collecting information on variants. So we're not collecting genomic data, but we're collecting information on genomic testing. And so where appropriate, we will create the fields and we'll store these data. Once we have the dictionary, we go through and we decide which are the most important things, which are less important. We do this coding, and then we have this bottleneck called harmonization, where we have to find a statistician to take the data and do this complicated mapping and to get it into a format that we can put into our commons. We're different than a lot of other commons that just say, give us all your data and we'll deal with it. No, we will spend a year, if needed, getting the dictionary perfect, having people harmonize their data, and then we'll do our quality control and put it into the commons. We want the data to be highly curated and pristine and ready for researchers. We've now done this over 10 different types of pediatric cancer, including cancer predisposition. We've had various versions of the data dictionary developed. We have contributors from all over the world. You have to come up with a clever name for your consortium, so we work hard on acronyms and initialisms. We form these consortia with memoranda of understanding that have to do with authorship and how you get data in and how you get data out. And now we're seeing lots of cases. We have over 30,000, 35,000 cases in the Commons, and over 30 papers have been published on data that have been collected for our Data Commons work. We work with groups from all over the world. We have, you can see highlighted in red here, are some of the groups we've worked with to get osteosarcoma data. We have over 26 data contributor and use agreements in place, and we're not scared of European regulation. We dig in. We have a full-time governance person that helps us get the regulations in place so that we can hold the data from any part of the world so far. We have not failed yet to get data into our commons where we've had a desire to put it there. We formed this Hibiscus consortium. You can see the chairs there. You'll recognize Katie on the list, as well as other folks that I'm sure you know, and we have groups from all over the world that are participating. This is the group that built the data dictionary. It's the group that's going to be responsible for helping us get all of these trials harmonized and into the commons. And there's a lot of data from these completed trials, and going forward, we hope to get registry data and other sources. And those of you who have access to sources of data, please let us know, because we'd love to talk about incorporating those data. If we get all the data promised to us, we should hit about 75,000 cases this year, but I think we're actually going to hit 100,000 because we're talking to other groups around the world with large sets of data, and once we can confirm the quality of the data, we really want to try to bring this in and try to make the data commons even more useful. We started as a collection of data commons. We had to do that because we tried to think about one giant commons, and then all of the disease groups sort of lost their mind. So we had to develop these individual data commons so we could assure them that we would have governance over each disease. But in the background, I've been thinking about this as one large data commons. There's going to be a single port of entry. The discovery portal looks like this. You come in, you can pick the disease you want to work on, or multiple diseases. You can select patient characteristics. You can even now connect to things like the genomic data commons, and you you can connect patients in our commons to patients in the GDC. This is publicly available. Anybody and your grandma can go to this and can do a cohort search and see how many patients are available. If you want the data, that's a different story. You have to apply for it and download it. The other thing we're launching now are visualization tools. So survival curves built right into the browser. We're piloting this for Rhabdo right now, and we hope to release it across the data commons in the next few months. And the last thing I want to talk about is our Gearbox platform, which we're super psyched about. The idea behind Gearbox is to solve this problem where you have a child who has refractory disease or is relapsing, and you don't really have access to understanding which clinical trials are available. And those of you who have gone to clinicaltrials.gov to try to find out the inclusion-exclusion criteria for trials, it's such a morass and a difficult system to use, and the inclusion criteria can be pages and pages long. So what we built is a system called Gearbox. We did this with funding from the Leukemia and Lymphoma Society to pilot in AML first. And what we do is we parse all the inclusion-exclusion criteria for all the trials available in clinicaltrials.gov, and then we present that back to the clinician in an easy-to-use interface where the clinician can then enter information about the patient, and as they enter the information, all of the trials will sort into match, potential match, and non-match. Now, you will say, well, there's other ways to do this. You could just download the trials and do it yourself. Yeah, I get it. This is like a really fancy calculator. But the whole idea is that as the patient is sitting there in the office, the clinician can go in and say, look, there's 4 trials. We're going to have extra information. We want to help to try to build a navigator system to help patients find out how to get on the trials. We're really excited about this platform. We just got a big set of funding from 2 different groups. One is a NIH-sponsored study, and the other is a foundation to expand this now across all pediatric cancer. So we're going to move from AML to support all different types of pediatric cancer, and we're very excited. Now, the next phase is not just going to be the clinician keying in values. We're going to be connecting to other groups like Tempus and Foundation for sequencing information and biomarker information. Hematologics does immunophenotype information. And Doug Hawkins doesn't quite know it yet, but even the COG we're going to try to hook into and try to pull data out so that it can automatically populate the Gearbox platform to help provide quicker matches for the patients that are being seen. So we think this has a lot of staying power. We think this is going to be a big thing, and we hope to implement it. It's now released in beta form for AML only, but it's for anybody in the US to use it. And I was surprised recently somebody from Amsterdam said that they use it during their AML tumor boards to help find trials for patients that they're talking about in their tumor board. That was an unintended positive consequence that I hadn't thought about. We have a team of about 22 people now. It's mostly data scientists and programmers and project managers who keep us on track. And then we have a full-time governance person. So this is expensive, tedious work, but we think it's really worth it because if you can build the right format for the data, then people want to put their data in there. And you have to build this trust and this friendship between groups to do this. So I'll end there with 2 seconds over, not too bad. And appreciate everyone's attention and look forward to the panel discussion. Thanks very much.
Hi, everybody. Brian Crumpton again from Dana-Farber. Whew, that was an intimidating talk to try to follow. I do want to say, when I first heard about what Sam was trying to take on, I was like, oh, that's never going to happen. And so it's pretty amazing, and I think unbelievably important for our field to have this data commons being built. So another thing I'm super grateful for. I want to talk a little bit about things that I'm involved with, but I'm really as a representative today in terms of how COG and Project Every Child sort of key into making big data. And I'm going to focus more on biology in terms of making big data from tissue that's biologic data. But I thought I'd take a step back and just make sure we're all on the same page about where that actually comes from. So there's a lot of ways to make big data from a tissue sample, biologic big data. You can actually make big data from one sample. If you do a complex enough experiment, you can generate an unbelievable number of data points from a single specimen, and single cell sequencing, for example, is pretty much big data even if you just do single cell sequencing on one tumor sample. You can also generate big data by doing simpler experiments on a lot of samples. And one of the challenges of trying to do that kind of work is to find enough samples, and there you really need multi-institutional collaborations to get the number of samples you want. Once you generate the data, it's pretty easy to share. Usually you can do it on an Excel table. and anybody can sort of reanalyze it, but it's not really that useful to reanalyze, because if it's a simple experiment, you already know what the interpretation is from the original person who did the experiment. Complex experiments on large data sets is really what we're aspiring to do, because that's where we're going to get a lot of information about osteosarcoma and understand the biology in a way that we really think we need to try to find better therapies. This is the hardest stuff to do, because you need multi-institutional collaborations to get the samples you need, but you also need expertise to analyze and reanalyze the data. But it's complex enough that it's probably likely multiple discoveries can be made on the same data set if you have people out there who know how to use it. And so one of the goals is to sort of lower the bar for using those types of data. One interesting thing, this is a plot from the Children's Oncology Group, just thinking about how do you get access to tissue samples from hundreds of patients with osteosarcoma. This isn't a slide about osteosarcoma, this is just about overall enrollment in trials at the COG level, regardless of diagnosis. But you can see that approximately half of patients who have a childhood cancer are being seen at one of 190 institutions, and only the other 50 are seen at the bigger institutions, a smaller number. But that's still, 200 and nearly 250 institutions. That's a lot of places to try to get samples from. And there's sort of two ways to go about doing that. One is that every patient who enrolls on a clinical trial run by a multi-institutional consortium, such as the Children's Oncology Group, usually there's a portion of that study where tissue is asked for, is either a blood sample or a tumor biopsy, usually a little bit of both. The benefit of the clinical trials is that they come with a very rich clinical data set, because as part of that trial, all the updates and all the scans and all the side effects from therapy are all being monitored. The other way to get samples is from patients who agree to enroll in banking. They may not be on a clinical trial at all, but they agree that anything left over after their clinical workup is done on their tissue can be used for research. And this means a lot more patients can participate, but it does mean usually that those samples come with a little less clinical annotations, because we don't know about every side effect and every dose of chemotherapy they've gotten. So those are kind of the pros and cons. But this is why the banking studies are so important. This is based on just SEER data, meaning registry data in the United States of the number of child-age people who get any sort of malignant diagnosis. And over time, because we've now developed more and more standards of care, a smaller and smaller percentage of patients with newly diagnosed pediatric cancer go on to a prospective study or a phase 3 study on COG. So this means, if this data is right, and I'm sure there's a big confidence window here, but assuming we're in the ballpark here of about 20% of patients going on prospective studies, that means about 80% of patients, we would not get access to the tissue without banking. And that's really where Project Every Child comes in. This is the biobanking study for Children's Oncology Group, and it's really the only way, honestly, in the world that you can get access to such large numbers of tissue samples from patients who want to participate and want to contribute to science and research. And these are some numbers that are a little outdated now, but I think from the most recent data pool from Project Every Child, they're just staggering numbers of 32,000 kids enrolled, 160,000 specimens collected, all of it annotated to some level, a lot of it with permission to contact for future studies, which means we can do a lot more things even than what wasn't necessarily envisioned in Every Child from the start. And it's a large proportion of the number of patients diagnosed with cancer during that period. So how do those samples that get banked become big data? And this is kind of what we're trying to overcome. Someone wants to do an experiment, they have to request the samples. So this is a picture from some random screenshot I took from the internet, probably copyrighted, I'm probably violating things, don't report me, of a DMV. So waiting in line, the line's moving so slowly that they put chairs into the line, right? So it takes a long time to get your questions answered. It can take a long time to get samples. Then those samples go to an investigator, experiments happen, it's sort of in a bubbly cloud. We can't do a lot to influence that, and then what gets spit out on the other side is data. And big data can sometimes feel like this large pile of papers that no one realistically can go through if they want to reanalyze. And this is really what we want, right? And we're not here yet. We may never be exactly here, but we'd love to be able to just go up to a kiosk and say, I need these samples with these genetic phenotypes, and because that'll fit because I can test my hypothesis with it. The investigator gets those samples, they do their experiment, and then the data goes into some, Iron Man type thing where they can like touch things, and new experiments and data gets generated, and an entirely new element gets created. That would be the dream. So how are we trying to make that more of the reality? So this is the Quad W office. The Quad W Foundation supports extra personnel within Children's Oncology Group that help make it easier for investigators to get access to tissue. Because of the level of annotation and the rarity of the sample sets, it is important that we vet every application. We don't want to be giving out tissue and have no one ever do the experiment, or useless data that's not even real come out of it. So we do vet this pretty hard, as does COG, as does the NIH. But this is a group that helps accelerate that, so that good projects get access to data, get access to samples and data a little bit quicker. And I can't really stress enough how important Quad W's funding of this office has been to accelerating research. That has resulted in a lot of groundbreaking studies, like the target osteosarcoma data set that's now freely available for any investigator to use and restudy. Germline studies one of my liquid biopsy studies was all possible because of the Quad W's office's improved access to the tissue samples in the COG. So that's how we're trying to improve accessibility. On the other end, trying to make data easier to use. This is a project that is led by Dr. Ji at UT Southwest. Her team is really expert in sort of creating user interfaces for complex genomic data. So this is the Osteosarcoma Explorer. Anybody with an academic affiliation can go in here and analyze the data. It's got all of the target data and a bunch of other stuff. You can do real-time hypothesis generating analysis of the data. We've even put our liquid biopsy data into this now from that previous study that I showed, trying to make it as comprehensive as possible. And this is a non-static platform where, as new genomic or complex data gets generated by COG samples, we can continually add those to this resource. A lot of people have visited this site. It's actually much more visited than I actually expected, because I didn't really even realize there are this many osteosarcoma investigators out there. But we've had almost 80 different investigators actually register for the site. And again, all you really need is an academic email, which is fantastic. We'd also like to enhance experiments, and this is a little trickier for us to do at the as a COG group. One of the things that I think probably a lot of you have heard is that there's this childhood cancer data initiative run by the NIH, but it's been a little disappointing because osteosarcoma hasn't been on the list initially for the genomic profiling return of results. But Corey's going to talk about the Count Me In protocol that she and Katie Janeway have started for osteosarcoma. And one of the things we're working on at the COG is to actually connect Project Every Child to Count Me In, so that patients who enroll in Count Me In, who agree to be contacted in the future for other studies, can be contacted by Count Me In, so that we can then do the profiling, and as part of that project, those results can get returned to patients. So I think that's one exciting way where we're actually trying to also improve access to, or actually try to facilitate important experiments being done. And this is kind of just a vision I put together of how we kind of think about making progress in the field as the COG and Quad W office. You know, patients contribute tissue, the COG gets that tissue, and then makes sure data related to it gets pushed out to SAM. Samples get pushed out to investigators, and in the future, Count Me In. And all that hopefully will come back together into a usable portal, and those get shared back and forth, and eventually discoveries get made that result in new therapies to try in clinical trials. And that's where Corey comes in.
Please don't start the clock for 3 minutes. Okay, sorry. Just want to thank the organizers here, Anne and Matteo, and the entire panel here for allowing me the opportunity to talk with you about this project, the osteosarcoma project, which is part of Count Me In. I know a lot of people here are probably familiar with it, but I'm going to give a little bit of background for folks who may not have heard of it before, and I would love to talk with anybody who's interested in this after the fact. So, back in the day, meaning 2015, the leadership at the Broad Institute of MIT and Harvard had this bold question. What if we could generate a massive publicly available database of clinical, genomic, molecular, and patient-reported data in cancer to enable researchers to find patterns in the data and help accelerate discoveries in the development of new treatment strategies? They brought me there because I'm a I'm a cancer research scientist, but I'm also an advocate. And I'm now, at this point, 12 years out from my own diagnosis of angiosarcoma, and so I understand the patient perspective to that, to whatever extent you can as one human. And together with Nick Wagley and the rest of the Broad, we built the Metastatic Breast Cancer Project, and we launched it in 2015. You can see here a map that looks like it's a bunch of people clustered on the East Coast, but I promise this is actually a representation of the population of the United States, and that we have over 6,000 of these people with metastatic breast cancer who have joined our study, making it the largest prospective cohort and big data for metastatic breast cancer that exists. There's been tons of papers that cite this data. We're writing our own position paper right now. And when the leadership of the Broad looked at me and they said, "Good job, what do you want to do next?" I just quietly said, "How about angiosarcoma?" Kind of turned around. And Eric Lander, who was leading the charge at that point, looked at me and he said, "Yes, absolutely, 100%. And I said, "Well, thank you." And so we did. And we launched the Angiosarcoma Project in 2017. And we have over 600 people who have joined. This is very similar in terms of size and scope to osteo. We have about between 300 and 500 people get diagnosed every year. Fast forward, we generated and released whole exome sequencing, tumor normal matched with clinical annotations, and we're able to make key discoveries early on that were published in Nature Medicine, just like Alana's paper yesterday. And the data there was also presented to the sarcoma community, who basically designed a clinical trial with the data kind of cast up in the background, even before it was published. And those trials are ongoing. Some are published, validating what we found. So we wanted to do that more and more, and I am 100% committed to doing that in rare cancers, and if I could do that in pediatric rare cancers, all the better. To the point of this panel, we look for everywhere we can deposit this data. So Sam and I are going to have a conversation after this, as well as everybody else on this panel, to make sure that we can be harmonized and out there in whatever way makes the data accessible. Right now, it lives in CBio portal and the genomic data commons that Sam was referencing, as well as Brian. And we also have our own Broad-based portals as well. Here's a face that is beloved by this community, Mr. Dillon, and he and his family and several other people, some here in this room, visited us as we were building the Osteosarcoma Project. And if you, I didn't get a chance to show you the websites from our previous projects, but they're very adult. And then Dylan and friends came and said, "No way are we gonna click on anything on that website. You have to make it way better and more fun and interactive, put videos on there, take away a lot of the words, and have it speak to me directly." And so based on a lot of conversations back and forth, we built and launched the Osteosarcoma Project. And we actually announced that at the last Factor conference back in 2020, I think, right before everything shut down. Since that launch, we've had 265 patients registered, almost 200 that provided consent. Several hundred surveys have been completed. We've requested several hundred medical records. We have 90 in-house. 4 tissue samples have been received, and we've received 84 saliva samples that have been subjected to whole exome sequencing, and they're being analyzed right now to look at the germline of osteosarcoma patients. But then, in the midst of this, there was an opportunity from the National Cancer Institute to write for a grant to bring a whole bunch of funding to some rare cancers. And not just some funding, but the ability to actually provide information back to participants in the form of germline and somatic data that was from both, their normal DNA as well as their tumor DNA. And we said, count me in. And we wrote this grant and we got it, which was awesome. And we used it to support the osteosarcoma project. So we launched it in 2020, but then after we were awarded this grant, we went ahead and retooled everything. And it took it's a big undertaking to go from research grade genomics to CLIA returnable genomics. And there are quite a few people in this room, chief among them, Alana Church, thank you, and Brian Crompton, who have been helping us along this process, along with Katie Janeway and several other people who've really come to live and breathe this project so that we could get this relaunched. That relaunch is happening imminently. And so anybody who has joined the project, if you want to rejoin, you can in order to receive results back. And those of you who have not joined, maybe in another week or two, you'll probably see an announcement through MIB so that hopefully you can also consider joining. Our goals are to generate a clinically annotated genomic data set. So this is going to be whole exome sequencing, whole genome sequencing, and bulk RNA-seq. And we want to share this everywhere we can. We also want to generate shared learning information, and I'll get to why that's such an awkward thing to say here in just a second. And we want to actually study the process of patient engagement so that we can learn how to do it better over time. And then we're also going to be collaborating with other centers who received a similar grant like this through what's called the PECGS, and that stands for Patient Engagement in Clinical Genomic Sequencing Network. I think there are 6 centers now. And it's really interesting. You can check it out if you just Google search PECGS. You can see some of the other rare cancers that are being studied through this mechanism. The areas that we have identified for impact, again, led by Katie and Brian, are really focusing on molecular characterization of different subtypes. I think we've seen quite a bit of data today to suggest that osteosarcoma is diverse, but they do break down into different subgroups. Look at tumor evolution. And this project is perfect for something like this, where you can get serial biopsies from a patient, but you can also collect serial blood biopsies from the same person as they go through their experience with cancer, and hopefully learn exactly what is going on at different time points as a result of treatment or progression and hopefully remission. And of course, the ultimate goal of identifying new targeted therapies, both liquid, this says rare. Oh yeah, so different types of tissue that may come online as a result of different surgeries and resections and biopsies that can inform on the disease. And then there are other benefits to participation, like learning about your tumor samples. This is as I said, we're now switching our gears so that we can give information back to patients. But we're not like a foundation medicine where we are designed to give them back within 3 weeks. So there's going to be a time delay oftentimes from when people sign up to when they may get that tissue information back. And so instead of calling it return of results, we're calling it a shared learning profile. We feel philosophically that if we can see your data, you should be able to see your data as well. And though it may not be as timely as something like foundation medicine, because it is going to be generated in a CLIA lab, it can be used by doctors to make clinical decisions. We also have a partnership with Invitae, so that if people want to provide a saliva sample, they can get their germline results back with a clinical geneticist in order to understand and interpret those results. This is a report drafted by Alana that we're calling Shared Learnings. And again, the goals are to learn and share information so that people can understand the value of their participation and understand a little bit more about the tumor that, that we're trying to learn about as an entire community. And again, germline results. So we will ask for saliva, and Invitae will run an 84 gene panel in order to see if there are inherited risk factors. So there are some challenges and lessons learned along the way. We have a low rate of return from medical records, and from networking and talking with different physicians, we've learned that adult clinics are different than pediatric clinics. So Katie is really helping us think through how we can directly connect with the right people at clinics in order to get the medical records in. There are challenges in acquiring tissue and challenges in developing this report in order to share information back to patients. So what's next? Relaunch, studying, and we're going to pilot a next of kin process that I would love somebody to ask me about when I have more time, because I just ran out of time. And with that, I just want to show a picture of the team. We have about 25 people that are working on this, and they're all glorious. So I'm excited to talk more about this with anybody who would like. Thank you. There's a bunch more slides. Somebody's probably going to have to forward through, but they're Patient Advisory Council, Scientific Advisory Council.
Next.
Clinical champions.
Great. My name is Elena Vasky, and I'm from UC Santa Cruz. And I wanted to start by thanking the organizers and this panel for having me here. It's truly an honor to be part of such an inspiring meeting and hearing all of the stories from the patients and the families. So today I'll talk to you about the utility of these shared data sets that we're creating and that we've been discussing in this panel, and hopefully convince you that yes, indeed, it is very important to share data and create these large data sets. Our initiative, the Treehouse Initiative, started because of a patient named Kelvin. He had a rare sarcoma, and he was actually from Canada. He was the first patient whose data were shared with our group, and some of the analysis that I'm going to discuss were done on his data and were helpful to Kelvin. And so we started Treehouse with this goal of applying these analyses, and I'm going to talk about the analyses and these large data sets to try to identify new treatment targets and to extract new knowledge for difficult to treat pediatric cancers. So our initiative deals with RNA sequencing data specifically, and we have this hypothesis that comparing RNA sequencing data from single tumors to cohorts of data can identify interesting genes, unusually expressed genes. We call them gene expression outliers, and can be useful for molecular classification of tumors as well. And this analysis improves with more data. And so to enable these RNA sequencing data comparisons, and I guess I should say why RNA sequencing? Well, it's because in pediatric cancers in general and osteosarcoma specifically, we know that there are defects in gene regulation, and we know there is contribution from epigenetic deregulations, and so looking at the RNA is important. And so to enable these RNA sequencing studies, we have been assembling data compendia of RNA sequencing data. This is a visualization of one of our compendium, and here every dot represents a single RNA sequencing data set from a tumor, and we have a variety of different tumors, both adult and pediatric tumors, that we have compiled from various different repositories. And so we have a number of these compendia available publicly on our website. We have compendia from primary tumors, from cell lines, from patient-derived xenograft models that Dr. Sweet Cordero talked about in the earlier panel. And so here are some of the sources that are included in our compendia, including the Target project and a lot of the resources we talked about in this panel and at this meeting, including individual laboratories. So we're very grateful to have access to all these data. So it's very important for RNA sequencing data to analyze the data in the same way so that we can compare it directly to each other. And so we spend a lot of time developing pipelines that we can apply to the data. And so we get raw data sets, and then we reprocess them with our pipeline so that the data are directly comparable and can be analyzed together. We also, because we get data from different sources, so we have developed a quality control pipeline that we use to make sure that the data sets, again, are comparable, and we sometimes have poor quality data that we cannot include in the compendium, and so we don't. And we do this by looking at this measure called mapped exonic non-duplicate reads, and if somebody is interested, I'm happy to talk to you in detail about that. So what do we do with these data? So one thing we can do with these compendia is we can look for gene expression outliers. So what this means is genes that are uniquely highly expressed in a given tumor compared to our whole compendium. So because we have so many samples, we can really spot things that are unusually highly expressed, and we call them outliers. And we can do this analysis across the whole compendium, or we can do it across a single tumor type, for example, across osteosarcoma or across a glioma, and we can find uniquely overexpressed genes in a given patient compared to other tumors. And then we can take these lists of outlier genes and look for pathway enrichment, so I identify signaling pathways that are overexpressed in a given tumor. And hopefully these pathways can inform us about what is driving that particular tumor and help us understand how to target that tumor. And so we have applied this analysis to several clinical cases and published a number of case reports describing how these gene expression outliers have been useful to identify a targeted therapy that was helpful. We can also mine these large data sets for novel biological impact, and so we've published a couple of papers understanding novel biology from mining these shared data sets. And I wanted to show you a case example of how this can be useful clinically. So this is a rare sarcoma case. Unfortunately, we don't have an osteosarcoma example, but I hope that next time we will have an osteosarcoma example. But this was a 1-year-old patient with myoepithelial carcinoma of the liver and was treated using chemotherapy and then had a recurrence. Because this is such a rare disease, there was no standard of care for the recurrence and no clinical trials for the disease. And so we did our analysis, comparing this tumor to our whole treehouse compendium, and saw that it's similar to some of the other cancers. So this is not really. So what I want to show you is this pathway, and then so we did the outlier analysis that I discussed, looking for uniquely overexpressed genes to reconstruct the signaling pathway that we think is driving the tumor. And so this suggested potential targeted therapies shown there, pazopanib and ribociclib. And in the absence, because there was nothing to try for this patient, so these treatments were tried. So pazopanib was tried first. There was some response, but then there was progression. And then later ribociclib was tried. And actually that resulted in a really strong clinical response. And so patient has been no evidence of disease for over a year, and so remains in that status, and we hope will remain in that status. So that's very exciting to us. And so we're excited to apply this analysis to more cases. So hopefully with this, I have shown to you that sharing genomic data sets can have real impact clinically and biologically. And our group does not generate data, so we rely on other people's data. And so we're compiling this compendia from other institutions' data and other labs' data. And so you can see how this is very useful. And I'm very grateful to all the investigators who have shared the data with us. Here's my acknowledgements. I want to thank my lab, the Treehouse Initiative, and our clinical collaborators, several of whom are in this room. So thank you so much, and our funders, and thank you all.
Did I push the wrong?
It's the big green button.
The big green. Okay, got it. Thank you. Well, I'd like to thank Christine and the organizers for the invitation to attend this wonderful meeting. It's really been great to be here. What I'm going to talk to you about today is the Children's Oncology Group repository and what we're doing for collecting cell lines and PDXs there and what we can do with them. But first, I'd like to dedicate this talk to Bob Seeger. Many of you know we just lost Bob last month. He was a mentor to me as well as a collaborator, and really the inspiration for my interest in working with a cooperative group and developing biobanking across the cooperative group. He was really a leader in that with neuroblastoma. This repository is funded permanently by Alex the Lemonade Stand, and we collected, as you'll see, mostly neuroblastoma samples into that, but generated a large number of patient-derived xenografts and cell lines. We also have, as you'll see, other types of cancers. And we distribute these widely, so there's literally investigators in 28 countries that are using these. The focus has been, for most of us, lately on patient-derived xenografts, but I want to spend a little bit talking about cell lines, because we learned so much from those as well, and I think they've gotten a bad rap that we need to reverse a little bit. Cancer cell cultures are really necessary for doing biological mechanistic studies. The preclinical therapeutic studies you can do with them can be done at a fraction of the time of a patient-derived xenograft study, especially for doing combination drugs. And there's data from just cell line studies alone that have informed clinical trials and led to phase 3 clinical trials that have been successful. And importantly, and this is an important component, and I think this will probably apply very much to osteogenic sarcoma, there are some cancers, particularly alternate lengthening telomere cancers, that just don't grow well in patient-derived xenografts, but you can get them established in cell lines, and then you can generate xenografts from those cultures. And the conditions you use for these are very important. And just showing you, this is from data on the neuroblastoma samples that we get from the COG, where we've been able to have enough sample to put them in various oxygen conditions. And almost 1/2 of these will not grow unless we use hypoxia. So the standard culture conditions you all are used to using of room air plus 5% CO2 would not grow half of the cultures that we were able to grow out from the COG. So for that reason, we use this strategy. When we get in a sample from the COG, we place it into culture in the standard culture conditions, so we have the comparator, but also into bone marrow level hypoxia and tumor level hypoxia, and also directly into the mouse. And we want to do these directly into culture and directly into mouse because we're trying to avoid multiple selection pressures, not to go through the mouse and then back into culture. So there's clearly differences between the cell cultures, the patient tumors, and the patient-derived xenografts. But we think by minimizing selection pressures that we're going to collect a set of samples that will be more meaningful by having it this way. And all these samples that we are able to grow then undergo STR validation against the original patient tissue, testing for mycoplasma. We rule out Epstein-Barr virus, make sure they don't have pathogenic viruses. And what's important about this collection is because the COG patient database is so rich, we have clinical annotation that is very rich on every single one of these models, and I get an update from the COG every 6 months, so including EFS, OS, the therapy that the patients have been on, et cetera. So I think that is one of the powerful things about doing this through the cooperative group, that collecting these samples that way gets us that kind of clinical annotation. Now, one of the important types of samples to collect are relapsed cell lines and patient-derived xenografts. They have a higher rate of growth in the laboratory than pre-therapy samples. And in fact, we have a 66% success rate of establishing, just from blood post-mortem, a STR-verified solid tumor sample, and not just neuroblastomas. We've gotten them from rhabdomyosarcomas and Ewing's as well. And these provide really ideal models to assess therapy resistance and therapies that might overcome that treatment resistance. And they've been really instrumental in establishing some molecular mechanisms of drug resistance that are not feasible with the limited patient tumor biopsy samples we don't get at relapse in our children with cancer. And particularly lately, we've been focusing on trying to establish these pairs of relapse and pre-therapy diagnostic samples. And those are, I think, going to be really useful for understanding molecular mechanisms of drug resistance and provide unique resources for various investigators for their research. Now, this is the panel of models we have right now in the repository, and as you see, the highlight is the neuroblastomas, where we have over 500 cell lines and 70 patient-derived xenografts. And the sad part of this for this meeting is that we have one osteosarcoma in each of those categories. And this is not because they don't grow, it's just that we don't get the samples. The neuroblastoma committee is really aggressive about getting the samples into the laboratory, and we just don't get them from the other diseases very much. And we're hoping to work with everybody in this room to really change that, because we think that these samples coming in, generating these models, are going to be useful for everybody in this room and everybody that's studying this disease. Now, just to show you the spectrum of what we have in neuroblastoma, these are the numbers of patient-derived cell lines and patient-derived xenografts by the various categories: pre-therapy, post-therapy, post-mortem, so in other words, we failed everything on this patient. We just didn't keep them alive. They died of their cancer, and we collected the sample, and we can work on it. But we have all the different biologies represented too: MYC amplified, non-amplified, ALK wild type, ALK mutations, and various different ALK mutations, telomerase positive, TERT promoter rearrangement positive, the alternate length and telomere positive models, then no telomere maintenance mechanism at all, even there's some of those. And now we're starting to accumulate genomic data on these through an R01. We've got whole exome sequencing, RNA sequencing, and with a collaboration with St. Jude's, getting this methylation profiling done. And because of dinutuximab being an important component of neuroblastoma therapy, now we're categorizing them by the expression of the antigen for that particular antibody. And interestingly enough, we find that these low expressors or negative expressors, not surprisingly, don't respond to the antibody, at least in our preclinical models. So I think that having this kind of wide number of models gives you this spectrum of having enough models in each of the categories to really do some research. And that's one of the things we think is of big value of the repository. Now at the end here, what I'd like to do is give you some examples of how these models have impacted what we're doing. This is a great one from Mayal Mosse's work where we established some PDXs from patients that had mutations that weren't as responsive to crizotinib as you would like. And you see there the little bit of response to crizotinib, but not much in the blue. But when she then treated this with the next generation ALK inhibitor, lorlatinib, we saw that this is her work, and from our models, that she saw this amazing responses. And that led to a phase 1 trial, and lorlatinib is now being, and I've never seen this before, but we're in the middle of a phase 3 study that includes crizotinib, and we're switching to lorlatinib so that we benefit the rest of those patients. And so every patient on this study going forward and this amendment goes through will get that next generation ALK inhibitor. And again, really this work came out of having the right models with the right mutations to study those drugs. Here's an example out of our lab, Balakonaru's article from this last August. He's here at the meeting if anyone wants to talk to him. Great study showing that ATM kinase is really a driver in alternate length and telomere neuroblastomas, and it's a driver of the drug resistance we see in those. And using a clinical stage ATM kinase inhibitor, he could reverse that resistance. And we went on from that to look at other types of cancers. And here looking at across neuroblastomas, rhabdomyosarcomas, colon cancer, osteosarcomas, lung cancers, triple negative breast, all that have the alternate length and telomere phenotype, you see the huge amount of ATM kinase activation that is across all of those. Just a comparison on the right side, you see the SKNB2 is a telomerase positive line. It has virtually no ATM kinase, but you hit it with radiation, of course it comes up. So a significant increase in this activated ATM we see in the alts relative to the non-alt models. And so we've gone in and looked at this, and that's the hypothesis from Sean Mecha, who just graduated with his PhD from the lab, and Bala. They said, you know, if ATM kinase is activated here, this is probably why these alt cancers so commonly defeat P53. They have loss of P53 function. What if you reactivated P53? So taking this APR246, a clinical stage APR, clinical stage P53 reactivator, we went in and then looked at these this variety of xenografts across histologies, and you see not much response to irinotecan in blue, a little bit better response to the APR246, but the combination of those are giving us these 100-day maintained complete responses in these xenografts. So we're very excited about that. You don't see this as you see in the right column, right panel there in telomerase positive models. And we did look at osteogenic sarcoma, at least in cell lines, and we see the same sort of pattern in a few cell lines and the same synergism with irinotecan. But we didn't really have the xenografts to work with, so we first looked at patient tumors with Tim Trish and saw that 74% of them are all positive. And then Phil Dela Cruz let us screen his models, and we've gotten some come in from him that are all positive. About 60% of his models are all positive. So hopefully in the future we'll show you some data in osteo with this drug. It's a very interesting drug. So in summary, patient-derived cell lines and xenografts provide a renewable resource, as Richard was saying earlier, for studying solid cancer, and I think that's important because you can renew it every time. You can grow more of them out. But it really comes down to more samples make more lines, and if we don't get them into the laboratory, they're not going to be available to people. So we hope that people will work with us on doing this, and it's important, I think, that all the COG cell lines and patient-derived xenografts are freely and widely available to anyone that wants them. We just put an EndNote library on there, which lets us anybody search by any cell line and find the papers that were published in it. And finally, the acknowledgments. This does not be done. I'm over time, but this would not be done without several groups of people: the collaborators, of course, the techs, and the students that do the work. But most importantly, I really want to thank the COG physicians, nurses, CRAs, parents, and patients. These are the unsung heroes. Without all the samples that you hear about, not only for our lab, but other people's lab, wouldn't be happening without them. Thank you.
All right, so it's hard to follow any one individual on this panel, let alone all of them, but I'm just thankful to the organizers and certainly to my other members of this panel. I'm very thankful to be here. Good afternoon, everyone. My name is Beth Stewart. I'm a physician scientist at St. Jude, and I was asked to talk to you today about our effort in generating the Childhood Solid Tumor Network. And so when I first started in the lab, there's actually a paucity of well-characterized preclinical models that were available. And in order to increase these efforts, we utilized a biology protocol that we called MAST, or it stands for Molecular Analysis of Solid Tumors. And we were consenting patients really for the past 12 years and collecting samples at the time when they would go for surgical resection. From this, we've generated over 300 PDX models from over 25 different histologies. They undergo the whole gamut of sequencing of both the patient tumor and the PDX. In terms of just osteosarcoma resources, we have generated 54 PDXs from osteosarcoma patients, 24 of those coming from we call the upfront or diagnostic samples, and then 30 of those coming from patients at disease recurrence. And I'll take a quick sort of opportunity here to also highlight other efforts. There's plenty of other groups here that have done PDX generation, and I'll highlight the PROC-C, which is a relatively new sort of collaboration of institutions focused on doing multi-directional sharing of PDXs and also biology studies and clinical trial development, you know, coming from, again, a number of individuals in this room that have been contributing to this project. I mention it here too because some of the early initiatives that are coming out of this are projects on osteosarcoma, which I think are incredibly important. So this panel is focused on big data, and I think Brian summed it up nicely of all the different types of components that be quote unquote considered big data. I'm going to talk to you today about some of our efforts in generating resources and data sets for the scientific community. And the first thing I'm going to talk about is something that I'm particularly interested in studying is disease recurrence. So as many of you know, a tumor is made up of not just the tumor cells, but also the cells of the tumor microenvironment. And we know that there's heterogeneity that's present. And it's not uncommon to see a number of clones that can be present in a tumor. Oftentimes you'll see something that looks like this, where 95% of the tumor can be made up of one major clone, and then there might be a minor clone that has a small percentage. And then at disease recurrence, we often see a shift where there's clonal selection, and you may have sort of that major clone sort of goes away. A minor clone now may become what we would consider more of a major clone or a bigger component, and then even have the development of new clones. And so we're currently performing single cell and single nuclear RNA sequencing to try to better understand the biology that's happening at disease recurrence and trying to make those data sets available. Sort of taking it a step further, to better understand what's happening inside the tumor with reference to treatment, I developed a biopsy protocol which allows me to sample the tumor from the PDXs at various time points, something that we're not really able to do in our patient samples or in our patients because of the inability to collect samples at so many different time points. And so from these biopsies, we can do a number of analyses, look at clonal evolution, look at the histology, do additional single cell. Some of the things I'm sort of showing some data here from one of my colleagues that did some work on rhabdomyosarcoma and learned a lot about sort of the developmental heterogeneity that's present in tumor samples. And so we're looking to expand that now to other tumors, including osteosarcoma. So I'd like to at least share kind of our next effort that I'm excited about is the iPSC generation that we're doing. I mentioned to you before that we have efforts in place to generate, PDX models from tumors that are resected from patients, but more recently we partnered with our surgical team to collect skin biopsies from these patients at the same time. And these samples are cultured in a dish, and these skin fibroblasts can be banked and later reprogrammed into iPSCs. From there, the iPSCs can also be banked and then saved for later differentiation into a variety of cells that make up the tumor microenvironment. Include NK cells, macrophages. There's a variety of protocols out there, to make a number of different cell types that make up that tumor microenvironment. And I showed you earlier that, our tumors, they're not just made up of tumor cells, but also these cells in the tumor microenvironment. And in some diseases, it's those cells of the tumor microenvironment that are sometimes implicated in disease resistance. And so by sort of combining these efforts, we can actually, generate a PDX, but also from the same patient, generate these iPSCs that can be differentiated into the cells where we can combine the tumor with the tumor microenvironment. Next, I'll talk to you about something that we call the Human Cancer Model Initiative. And we've been working to utilize our PDXs in sort of a number of different ways. And I think also mentioned from Pat, having the ability to do in vitro analysis is really helpful. As you can see here, we have sort of a schematic of how we're generating these, what we call 3D or 2D models, which are spheroids. And so you can take a PDX tumor, dissociate it, put it on a Lipitor plate, and they'll form sort of these spheroids or 3D looking models. Then if you transfer it over to a Geltrex coated plate, they'll sort of spread out into more of a 2D system. We're doing characterization along the way at each one of these steps, and then also doing reimplantation along the way back into the mouse to make sure that all of these different components can go back and form a tumor. And I'll show you sort of a picture here of kind of what this looks like. On your left is just like one of these spheroids look like all the cells sort of are clumped together. And then there's a picture of sort of a multitude of spheroids together in a dish. And then one of the things I think is really important is maintaining tumor heterogeneity. And so as cell lines are incredibly important for a lot of the work that we do, there are some things where maintaining that heterogeneity also could be very important. And so this is just sort of an example where we compare sort of patient and spheroid and are able to maintain the heterogeneity within these model systems. And so I think that goal of generating model systems that can maintain this are certainly helpful for a lot of the biology that we want to look at. All right, and the last thing I want to talk to you about is the Childhood Solid Tumor Network, and this is something that I'm incredibly proud of. I think if you look around this room, the number of people that are sharing resources, I think, is just phenomenal. And so this is one of the things where we want to share not just resources here within the U.S., but really throughout the world. All of the PDX models, the data, iPSCs, the 3D, 2D model systems, all of them are made available to the scientific community through the Childhood Solid Tumor Network. It's free of charge. There's no obligation to collaborate, or you don't even have to give us any credit. We're just thankful if people are able to use them. And thankfully, many people have also contributed resources. To date, we've had over 575 requests. Sent samples and data to over 200 investigators at 110 different institutions. It's, again, something I'm thankful, but I didn't do this alone. It's a collaboration of a lot of people working together to make this a scientific resource that I think is heavily used, and very thankful to have it. So with that, I'm going to just turn into a couple future directions, and I'll go through this quickly, but certainly one of the things that we want to do is continue to use these model systems to better understand, what's happening at disease recurrence. This is what's taking the lives of our patients, and so can we better model the tumor microenvironment and try to better understand that, both in vitro and in vivo? Expanding single-cell sequencing efforts and making that data available, looking at heterogeneity and osteosarcoma and other diseases. Doing comprehensive clinical testing, preclinical testing to get the right drugs to clinic as fast as we can through efforts like the PROC-C, through Pivot, and a variety of other people that are doing this work. And then expanding, and refining. How do we share big data and foster collaboration? I know people sort of alluded to this, but I share this example of our COMET project. We are launching a project now where we're trying to do DNA methylation on over 5,000 pediatric solid tumors, includes over 500 osteosarcoma samples. And there are many of you in this room that have generated samples and sent them to us for this analysis. We're going to be excited, hopefully, in the next year to make this data available for everyone to go mine. Not only will it just be methylation data, we're also trying to collect slides and take pictures, IHC, a variety of things where you can actually go and mine the digital sort of images, trying to pick up different features of these tumors. So something that we hope will also be a nice resource for the scientific community. With that, I'll wrap it up. Just want to say thank you specifically to the organizers of this conference, but also to our patients and to their families. Thank you.
So incredible group of talks. Thank you. Thank you for all the important work. I think we can throw it open to questions. but I'm going to look quickly, and I think they're grabbing questions, so I don't want to jump ahead of those. Are there questions?
Any questions out there?
So I'm going to start.
I do have a list of
Okay.
questions as well.
Okay, all right. So go ahead, please.
I have a question regarding tumor microenvironment. Is there any chance that we can overcome not having so that the murine microenvironment doesn't take over the human tumor microenvironment, which is actually put in to start with, and how do you think that that will impact results?
Yeah, so, I don't know if this is on. Can you all hear me? Okay. So it's a great question, and something, like I said, we're excited to start doing. As we've generated these iPSCs from patients, we're going to be making these components of the tumor microenvironment. I hope to be able to put the same PDX with those cells in the tumor microenvironment back into the same mouse, but also sort of partnering with that, there's all these efforts in making humanized mouse models, right, trying to overcome some of this as well. We are a little bit, away from getting to this true, I would say humanized model where you would feel comfortable fully testing immunotherapy and a variety of things like that, but I'm hopeful that that's in our future. I'm excited about this work. I think that's why also having things like these 3D, 2D models where you can do some of these experiments in a dish first, where you don't have that sort of mouse component, might be a way to sort of get around some of that, at least in the interim.
Thank you. I have several questions I'm going to pose to the panel, and this is coming from a mother. For Dr. Painter and others, do you see the Count Me In project adding data from liquid biomarker of circulating tumor DNA?
Hi, is this on? Can you hear me?
Yes.
Yes, absolutely. We do see that, and we are currently deploying blood biopsy kits for patients to take to their normal treatments. And if they're not on therapy, then we put a Quest voucher form in the boxes so that they can schedule an appointment. But that is something that we are very much interested in. And I'm actually going to turn it over to Brian, who is going to be leading a lot of the analysis work from what comes in from those blood biopsies.
Yeah, I think one of the tricks for it will be to figure out what the meaningful time points are, since it's not a clinical trial where we know we can get very structured time points on the blood draws. Corey and I were actually talking earlier about, thinking about studying around the time of new diagnosis and then at relapse to try to really understand tumor evolution, which is completely unexplained, I think, because we rarely do biopsies at the time of relapse. We know what it is. It doesn't help patients clinically to get a surgical biopsy at that time because it doesn't inform treatment yet. And so, but blood collection is minimal risk, so we can certainly do that. So I think trying to make sure our time points are what we can get within Count Me In and then use it to make a real discovery will be important.
Thank you. And again, for the rest of the panel, or for the entire panel, are surface protein emissions data used for prognostic biomarkers? And if so, is it a strong metric?
I am the leader of the panel, so I'm going to sort of defer to the rest of the panel. With Brian trying to get
Can
me
we
to answer
defer
the
back
question.
to you, Richard?
I think so far some of the earlier sessions covered it. There aren't many biological prognostic factors, unfortunately, and that includes surface markers, meaning the most promising that sort of seems like it's sort of emerging is probably MYC amplification as a biological marker that seems to have the most validation. There have been other markers that have been looked at, like HER2, but generally they haven't been reproducible. So the only real clinical prognostic factor we have for osteosarcoma is the presence of imaging metastatic disease. So unfortunately, that's all we have at the moment.
Thank you. Oh, I'm sorry, go ahead.
I was just going to say that, Richard just published a study on the surface expression of proteins in osteo, and I think we don't have enough clinical data yet to know that any of those are not. And I think it's also really important to think of biomarkers in the context of therapy, right? So a biomarker of successfully surgical excision would be, are they in places where you can surgically remove them, right? But if you have a therapy that targets a surface protein, then of course that surface protein becomes the biomarker for that therapy, especially if the therapy is effective. So it'll be a moving target for hopefully the rest of our careers, where more and more things become biomarkers because we have more and more targeted therapies.
Thank you. And one last question from the list, and I think this might be for Dr. Bolkenbaum. Are there biobanks collecting info on immune responses of cancer patients to create an ideal immune profile?
Yeah, I mean, I think other folks here probably know more about this than I do. So, most of the biobank that we link to directly is the COG biobank in Nationwide. So we can tell you for any particular set of patients exactly which patients have samples. The immunophenotype data we're collecting right now is mainly for AML patients through our work with Leukemia and Lymphoma Society. But if anybody is aware of a registry or any sort of repository of data, we can actually take that immunophenotype data and link it back to the clinical data that are collected on those kids and hopefully to the other data that are collected. But I defer to the other panelists if they have other info about that. I don't.
Thank you. Any other questions?
Yeah, I see questions on the other side of the room.
Okay.
But I'm going to take the break while you walk over to ask the question I was going to before. So you guys have talked a lot about data sharing, and it's clear that's a big part of what you guys are committed to, data sharing, model sharing. You know, there's a lot of people doing this, and how do you sort of measure commitment? How do you get a sense whether, like, these different groups are as committed? Like, what do you do to show that you're fully committed to this part of it that we could use as a bar for others, perhaps? Just anyone's thoughts on that?
I'll kick this one off. So for us, it's always been a coalition of the willing. And we, if you look at data sharing initiatives across the world, they are often rooted in some sort of monetary incentive, you know, whether it's collecting data to sell back to pharma or to use for synthetic controls. We have found that in the pediatric community, we have had no problem getting involvement of people who want to participate. In fact, our problem is that we have a lot of people that want to submit data that are non-traditional data submitters, and so we have to talk about things like data quality and where the data come from. But I think that for us, the idea is always to make the tent as big as possible. So I love it when somebody says, oh, we can't work with Dr. So-and-so, they're really hard to work with, they're our biggest competitor. So I say, the next thing we do is we write an email to that person and say, you gotta work with us. So we make the tent as big as possible, and we've never, ever had a problem. We've never had somebody come to us and then say, we're not gonna give you our data because we're worried about X, Y, and Z. I think, and I didn't have time to go over the one slide I have, which is all about, it's about sharing, it's about, I mean, it's pediatrics, right? It's about sharing, it's about trust, and it's about letting people know exactly how the data are being used and what they can expect from it. So that's my perspective from the PCDC, but I'm sure others have sharing perspectives as well.
Yeah, I'm going to share something that I would have never guessed was a barrier to sharing data, which is how hard it is to share data. It is not trivial just to give data away. It is really challenging. We have a team of about 5 people and it took them about 6 months to figure out how to get data into the Genomic Data Commons. And they didn't necessarily want to take it because it wasn't NCI funded. And so there are all kinds of, you would be shocked at how hard it is to actually share data. And I am routinely asked by others that are in the nonprofit sector that haven't gone through the process, hey, what do you do? Who do you call? I'm like, oh, friend, come sit down. We'll have a conversation. And so I just wanted to put that out there because it's something that caught me by surprise. With Count Me In, we leverage the will of the patient in order to liberate data. And so we have the patient's consent in order to get medical records and make that publicly available, as well as acquire tumor samples. And so once it's in our hands and generated, we can and we do make it publicly available. But some of the barriers that we found are that some individual institutions are reluctant to give us the tumor samples or the medical records without some promise that they will have intellectual oversight and property over anything that comes downstream of discoveries that are made, which is problematic. And I think one of the ways to get around that is just to shame them, from the perspective of advocates. And so, though I can't sit up here and do that publicly, if anybody wanted a sidebar with me over a drink later, I'm happy to give some stories.
I say I'll add on to that too, just sort of the flip side of it. In being, someone who prefers to share as much as we can share, I've never been in a situation where I have felt scientifically scooped or taken out because I have been willing to share. And so if that's any bit of incentive for people, I would like to think we're in a like-minded community here of respect in individuals, and I've not found the flip side of where there's been a downside to sharing.
Yeah, absolutely. I agree with that as well. In this pediatric community, we also have had, everybody is really willing to share, patients and labs and institutions. I would like to agree with Corey that it is incredibly difficult and it's also expensive. And it's not, people often don't understand, don't recognize that it's expensive. We also have a full-time data coordinator kind of hunting for data and also dealing with data use agreements, because every site, every laboratory will have their own agreements for good reasons, because this is sensitive data and raw genomic information is identifiable information, and so there are restrictions around it. But so dealing with all of that is both expensive and hard, and I think that's not appreciated for those who don't know.
I do want to add on the quick point, though, that we have built an incentive structure in academia around tenure that does not incentivize data sharing like we're talking about. And I'm not aware of anybody who's been promoted because they were the best sharer of data. And it's funny to say, but there will come a time when those who create better data sharing systems will actually be seen as contributing academic progress. And so there's a natural tension there. And as those of you who are raising funds and creating funding opportunities, I encourage you to put very very strongly worded sharing requirements in your RFPs that require the sharing of the data after some embargo period. Don't shy away from that. It's incredibly important to do that. And the NCI has not taken a front seat to doing that. And so I think they need to follow, and I think you guys need to set the example.
So I think this is brilliant, but I heard something that I think is really interesting. I think I heard the word pediatrics like 80 times. And the adult medical oncologists practice differently than you guys do. And so when you ask the question, do we biopsy first met? I have like 10,000 reasons to biopsy the first met inside a 70-year-old smoker. We have these banked. You want to ask these questions, you want to do big data, don't forget about us. We might have what you're looking for.
Great point by Brian. Does anyone want to tackle the inclusion of adults in your particular initiatives? Is that something that's possible, not possible?
I'll take that.
Everyone grabs the microphone.
We enroll, we now have our, the Count Me In program is open to all cancer patients who have ever been diagnosed in the United States or Canada, regardless of age. So we have people that are less than a year to people that are 94 years old. And we do have serial biopsies from some of those adults. So thanks, Brian.
Yeah, I think the biology power of having all of those paired samples is incredible. And I'll just speak to our COMET project of methylation. We are including adult tumors as well for a comparison. So appreciate people that have contributed those samples to our project as well.
So we also include adults in Treehouse. It's much easier to find adult cohorts. So we spend a lot of time looking for pediatric, especially like rare tumors, but we definitely have adults. And it's, there is many more FDA-approved drugs for adults, and so situations where you're looking for drug repositioning, it's very important to include adults so that we can see instances where there could be a common target with the adult tumor.
Not only are we taking AYA in young adult patients with pediatric diseases, we are now moving into adult cancers as well, because the same techniques we've developed, of course, work across other rare diseases, and so we're expanding our commons. We're going to the Pediatric Cancer Data Commons are going to have to undergo a rebranding in the next year because we're obviously not just going to be doing pediatrics. I will say that working with the adult consortia has been a challenge. The pediatric community is I have found very easy to work with, and once we start including SWOG and ECOG and Alliance and Energy and these other groups, it becomes increasingly difficult to convince them to throw their data into the mix, but we're getting there, and we're doing it by pushing with the pediatric diseases like Hodgkin and germ cell tumor and moving in that direction.
I think one of the other challenges is that for a disease like osteosarcoma that's primarily seen as a pediatric disease, although it certainly affects adults, it's not like our adult colleagues are so plugged into Project Every Child and COG, so like if you don't have a place to put a sample, like it just goes into a pathology freezer, which is, great that it's not getting thrown away, but it's not necessarily something that is so easy to track down and doesn't necessarily become big data and get funneled in. But I think all of us are super interested in trying to break that down. I mean, to me, Count Me In is one of the easiest ways to break that down because it doesn't require, honestly, I'm obviously biased, so, but it doesn't require someone to open Project Every Child for perhaps what accounts to 1 or 2 patients over a 5-year period, which is very expensive. Patients can just come to it themselves, and I think that that's sort of the power of the Count Me In project.
I do want to amplify Dr. Van Tine's comments, though, briefly. If you look at the preponderance of oncologists here, it's mostly peds oncologists with important representation by Dr. Van Tine, Dr. Fetterman, and others who are medical oncologists. Again, mutual understanding is always fostered by being together. Maybe as a sort of tangible note, we can sort of try to draw in more of the medical oncology community. It's worthwhile. Obviously, the group spans both, peds and adult age ranges. So great question and great comments.
Any other questions from the audience?
So the inclusion of adults would be one answer to drive up volume when we're looking at measuring success in dozens or hundreds with how rare this disease is. How would you characterize the present and future role of synthetic data in looking at new research insights?
I love this topic. Thank you for bringing it up. I think that whenever you stray into anything rare and clinical, you have to start paying attention to what you can do in terms of building real-world data and transforming that into real-world evidence. I know that there's the FDA is really interested in this right now and recently formed like a nucleus to really deeply explore this. And so just know that the FDA is pushing pretty hard from inside to give good and as clear guidance as the FDA can on how you can build some of these data sets to be considered for evaluation if you're trying to build an arm. And just, to maybe just expand on it a little bit, if you think about trying to make clinical trials in rare patient populations, you can't accrue necessarily the amount of people that you would. And sometimes when you have a rare cancer and there's a hit or a target, there are multiple people that want to develop drugs and it dilutes the patient population. And so there's just all kinds of issues and barriers that come when you try to develop drugs for rare cancer patients. And if you can do this thing where you are able to use real world data to generate a synthetic control arm, you can largely eliminate some of those barriers. And so I'm a big fan of that. And we're working right now with the CPATH Institute, and they work closely with the FDA in order to do something like tangential to this that I'd be happy to talk with you offline, but it has to do with drug repurposing and real world data.
I'd like to add to that. Let's not forget that the guidance for the FDA issued on approvability of 2 novel investigational agents allows you to use non-clinical data to support your approvability. That came out of a PZODAC meeting we had where we said it just wasn't ethical to require that we did these single agent trials. So I think the non-clinical data in the PDXs can contribute to this.
For sure.
And there's a group that just not for profit that just started this last year called the Clinical Research Data Sharing Alliance, which I'm a part of, and it includes members from CPATH and Project DataSphere and other groups. This is a huge topic, the idea of developing synthetic controls and real-world data and real-world evidence. And so, if anybody wants to talk more about that, there's tons of information out there. It's not trivial, it's not easy, but it's going to be essential going forward to do these kinds of studies.
So I wanted to ask, it's sort of divergent elements. We've heard lots about PDXs from all of you and know about it, but what about orthotopic PDXs and how important are they? And here Dr. Hoffman, who's from San Diego, is a great proponent, for example, of that in osteosarcoma and others. And in the other direction, organoids. And so we saw some spheroids presented in some pictures here today, but I heard nothing about organoids from any of you. And I'd like your opinion relative pros and cons of these.
Sorry. Beth and Patrick.
Yeah. Well, I constantly hear people, and at least I'll just go to neuroblastoma because that's what I mostly talked about orthotopic, xenografts. And I said, where? Okay, we don't worry about the primary tumor. Okay, that's not what's killing these children. It's metastatic disease. So I'm a little dubious on the importance of that. Now, osteo is a different kettle of fish, and I won't comment on that. I'll leave that to the osteo people. To answer your question on organoids, basically what we grow in culture is essentially a combination of some cells attached to plastic and a bunch of balls that are organoids, and that's across all pediatric solid tumors when we get them. So we crop preserve early passage material, and we carefully select so that we're carefully not select for attached versus floating and spheroid collections, so the heterogeneity is preserved there. And so yeah, you hear a lot about organoids, but essentially what I'm talking about when I talk about patient-derived cell lines is a combination of everything, okay? Which I think is better than just selecting for the things that are floating in balls in the dish by a special mechanism to make them float as balls.
Yeah, I think we could probably debate orthotopic versus non-orthotopic models for quite a while. I think it also depends on what you're trying to do and what you're trying to study, and so doing it in context. Certainly if you're wanting to look at the tumor microenvironment or some of those components, having an orthotopic model might be helpful, and so that's why that's been an initiative, at least, that we've had at our institution. In speaking to the sort of organoid versus spheroid, to be honest with you, I think there's a spectrum of what people call these things in the community, whether or not it has a stem cell or not, that's where it really is the defining moment. And so I'm cautious when I talk about our models for these pediatric cancers, and I've been calling them sort of 3D, 2D spheroid type models, because are they truly what we would call an organoid? I think that's, again, to be debated, but some would take these exact same models and call them spheroids.
So we're coming to the end of this panel's time. I really want to end by thanking the panel both for their great talks as well as for their contributions both individually and collectively to sort of advancing this whole concept around osteosarcoma research, data sharing, material sharing. So thank you for everything you guys did in your talks.